All right, welcome to the third lesson of the PopGen and Comparative Genomics channel. I'm Jake Weissman. I'll be teaching this lesson on phylogenetic structure, what it is and how to correct for it. This is going to be kind of the first lesson dealing with the comparative part of the channel. We're not going to talk about PopGen so much today. Most of this lesson is going to be kind of redundant with the text lesson or the text markdown lesson that will be posted. And so if you don't like watching videos, you can just search that and read it. But this is for the people who prefer a audio and video recording. So I'm going to share my screen. All right, so I've given it this dramatic name, but really we're going to talk about phylogenetic structure and what that means. And so to kind of put this lesson in context, we've been talking about population genetics in the last two lessons where we talked about tests for selection. And in population genetics, you're usually looking at variation within a population, thinking about genetic variation typically. And then by assessing those patterns, you can say something about the evolution of that population. When we're thinking about comparative studies, we're often looking at large sets of species and making comparisons between those species and looking at their phylogeny to make conclusions about evolution. So things like the types of questions I'm going to ask are, do sets of traits tend to evolve together? Are there traits that affect the rate of diversification along the lineage? So, right, do wings in the insects, for example, lead to a burst in speciation? And then thinking about kind of going back in the phylogeny, can we predict what things were like in the past, right, from current states? And so we're really going to talk about the first question today. And to give you an example of kind of the types of questions you might ask, so I'm a microbiologist. I don't usually study pathogenic bacteria, but say I want to like get an NIH grant, so I'm going to all of a sudden become interested in pathogenicity. And so I want to say, okay, what traits of a species are going to be associated with that species being pathogenic in humans, or maybe in plants or some other species? And maybe for some reason I am convinced that genome size should be associated with pathogenicity. I don't actually know if this is true. I just, I'm thinking of two traits that people might be interested in. So I want to test the hypothesis that genome size is associated with the evolution of pathogenicity. Kind of a way of testing this hypothesis is to look for a correlation between genome size and pathogenicity across species. If I do that naively, just calculating correlation, or just doing like a linear regression, that's actually going to lead to some problems, right? So statistically, that will be a bad approach. And so I want to tell you why that's going to be a bad approach and give you an illustrative example. And so the problem that's going to lead to all of our woes is that there's hierarchical structure to our data. What we're calling phylogenetic structure, right? Because the hierarchical structure is the history of past species interactions. And this means that our data points are not independent. And so a lot of what I'm going to talk about today comes from this paper that Felsenstein 1985, where the idea of independent contrast was introduced. And basically most of this lesson is really just a rehashing of that paper. It's a very clearly written paper, and I recommend reading it. It's a very good read. It's a classic text in this field. And so to kind of get a sense of what we're looking at, why don't we think about a phylogenetic tree and what they look like? So hopefully you've seen a phylogenetic tree before if you're at this lesson, but this is a simulated tree that I generated in R. And each of these tips in our data set will be a data point, right? And we can say, okay, each of these species, maybe they're species of bacteria, and we know whether they're pathogenic. Say we already know whether they're pathogenic and we know their genome size, right? And so there's 1,000 tips on this tree. We have 1,000 data points. That's plenty of data, you would think. But if you actually start looking at this tree, you might realize that there's some interesting patterns, right? So these different species have been evolving independently only so far back as their most recent common ancestor. Before that, each pair of species was the same lineage, right? And so if you pick a tip all the way at the top of the tree and all the way at the bottom of the tree, those tips have been evolving independently really since the very root of this tree. But if you pick two tips kind of up here, they've only been evolving independently for a very short period of time, which means that in terms of their traits, they will have only diverged a tiny bit. So when you treat them as independent, you're actually double counting all of their shared phylogenetic history. And so what happens, because there's two big clusters in this tree, and most of the evolutionary history happened before kind of these clusters really started splitting off more, right? So most of the evolutionary history is along these two long branches. When you start counting all these tips as independent, you're basically counting these long branches repeatedly, a thousand times, basically. And we call this pseudo replication. It's basically artificially inflating your data set where you think you have more data points than you actually do. And if you were to simulate two traits on this tree using I'm not actually going to get into the model right now that we use to simulate traits, but if you use kind of the simplest model to simulate the evolution of two traits on this tree, you might end up with data that looks like this. And so I've simulated two traits. I'm calling trait 1 and trait 2. You can imagine them as pathogenicity or a degree of pathogenicity or something, which isn't really a continuous trait, so maybe it's a bad example. But genome size is kind of more easy to understand in this context. And so I let two traits co-evolve, two traits kind of evolve on that same tree, but with no correlation between them, right? So these traits have no actual association with each other. They've just evolved on the same tree. But if you look at their kind of correlations, just their general correlation, they are highly correlated because they have this shared evolutionary history for most tips in this tree. And so you get a highly significant result, even though there's no actual link between these traits, right? And so this is kind of the worst case scenario in statistics, where your methods are going to lead you to have a highly, highly confident spurious result, right? Because your p-value on this regression, looking at these confidence intervals, is going to be, I don't know, probably below machine error in R. So you'll get that like 2.2 times 10 to the minus 16 if you see it in R, where it's basically just saying that you're basically at a p-value of 0. And so the moral of this story is that phylogeny matters, and that if you don't account for dependency structures in your data, you can get spurious results. And these spurious results will look really confident. So you'll think you have a really strong result, even though there's absolutely no pattern. And so it's really important that you correct for these patterns. And so I'm going to take you through a tutorial in R that will allow you to do this. And so I guess often we do these lessons in two parts, but we can probably just put them all together. I'm going to unshare this screen for a second. So associated with this lesson, there is an R tutorial written in R Markdown. The first section of the tutorial is really just a rehashing of what I just said, with some more, there's kind of more details. There's some quotes from the Felsenstein paper. I have a lot of links, including links to some free texts that might be useful to you, especially if you want to get more in depth in these types of approaches. And then I basically walk you through the same example I just took you through, but with more instances simulated. So I have 9 versions of that tree simulated, and then I've simulated traits on that tree again. You can see that each time you simulate a different tree in a different data set, you get a different but very significant pattern. Whereas if you looked at a tree that had no hierarchical structure, so there's a star-shaped phylogeny, where there's no dependency structure here. Each tip has kind of evolved along its own branch. And so where you have no phylogenetic structure, you get no spurious result, right? So you can see these are all flat, even though I simulated the data in the exact same way. I give you a few more details on how this data is actually simulated and what those models are. I also talk about providing some intuition in terms of pseudo-replication by making a comparison directly to just replicated data and what that can do to your data. And then I give some more kind of simple examples to build intuition. But the part of the lesson I really want to kind of emphasize is that there are methods to deal with this, right? So I kind of gave a very hopeless result in the example where you got this highly confident but spurious result. And so What can you do? Well, the first thing that people often want to do is test for phylogenetic signal. So if your trait evolves extremely rapidly, phylogenetic signal may actually not be a problem, right? So if your trait evolves so quickly that all history becomes sort of irrelevant because that history is kind of washed out by new changes, then you don't have to worry so much about phylogenetic signal. But if you have a very slowly changing trait or slowly evolving trait along the trait, then you have to be a little bit more worried. There's ways to test for kind of how much phylogenetic signal is in a trait statistically. Um, I give you an example of two using the phylosig function in the caper package. And so one's a non-parametric method that this Bloomberg's K, where you basically shuffle the tips of the tree. And then there's Pagel's lambda, which is a more traditional method that is parametric, I believe. I need to go back and read that. Phylogenetic signal only gets you so far, right? So you know that something's wrong if you have phylogenetic signal. Well, not wrong, but you know that you have to be careful if you have phylogenetic signal in your trait. But just knowing that doesn't allow you to necessarily do anything about it. But there are methods that allow you to account for phylogenetic structure directly in your statistical analysis. And so I talk about the phylogenetic generalized least squares method, or PGLS. And the way PGLS works is that basically, if you have a known phylogeny, you can take that phylogeny and encode it directly in the covariance matrix of a generalized least squared model, or a generalized linear regression. And this is kind of similar to like a mixed effects model for people who've used that before, where you have some sort of dependency structure or some sort of structure to your data that you're encoding in your covariance matrix. To get from phylogeny to covariance matrix is not a trivial process. You actually have to specify a model of trait evolution, because you need to know basically how does a phylogeny translate into dependencies between traits and between tips of the tree in terms of that trait and between the two traits. And then to do that, you kind of have to know how they evolve along the tree, the different traits. And so we are going to have to assume particular evolutionary models. The simplest evolutionary model is a Brownian motion model, where you kind of assume just random drift along those different branches of the tree. But there's also the Ornstein-Uhlenbeck model, which is another common model where you're basically modeling that same drift, but it's essentially Brownian motion on a spring, where you have some optimum that you're being pulled towards for a particular trait. And then I give an example of how if you misspecify your model, so say you have a trait that's evolving like by an OU model, but you use a Brownian motion model and you did your statistical analysis, you can also end up with a spurious result. So I give an example of how that can happen. And so these methods are, a little bit finicky, right? If you have the wrong underlying trait model, you can get the wrong result, and you can get a highly significant result when it's not actually real. So I often will try to apply multiple models, right, and say, well, this was significant, taking into account various different evolutionary models. So that's one approach. There's also a lot of newer literature that's talking about rate shifts along the tree. So if your trait evolves faster in one part of the tree than another part of the tree. So I guess an example are like if you're familiar with like the Lenski experiments, if you have like a mutator mutant where you're going to have a much higher mutation rate, then you might also see faster trait evolution. And so you'd have to account for that in your model. So there's models, there's newer developments in terms of methodology that allow for that. Most of these are implemented in R because this community tends to be pretty R focused in general. And then I give you a couple links to like other types of comparative analyses that you might be interested in that maybe we'll cover in future lessons. So yeah, so without getting too much into any of the math or details statistically, that is a brief introduction to phylogenetic structure.